In a hospital in Guangzhou, China, a 39-year-old man lay brain-dead from a hemorrhage. What happened next will be remembered as one of the most significant moments in medical history. Surgeons replaced his left lung with one from a genetically modified pig. For 9 consecutive days, that pig lung kept working inside a human chest, breathing life into the future of organ transplantation. This isn't just another medical breakthrough. This is the moment we proved that the organ shortage crisis killing hundreds of thousands of people every year finally has a solution. Welcome to Curiosity AI Podcast, Episode 18. I'm your host, and today we're examining a medical achievement that sounds like science fiction, but represents the cutting edge of genetic engineering and transplant medicine. On August 25, 2025, researchers published results in Nature Medicine that stunned the medical world. For the first time in history, a pig lung successfully functioned inside a human chest for over a week. This wasn't luck or a medical miracle. This was the result of years of precise genetic engineering using CRISPR technology to create the most human-compatible pig organ ever developed. But before we dive into the technical details, let's understand why this matters so profoundly. Every year, 173,000 organ transplants happen globally. That sounds impressive until you realize millions more people die waiting. In Europe alone, nearly 4,000 patients are on lung transplant waiting lists. Every day, people die not because their condition is untreatable, but because there simply aren't enough donor organs. Lungs present the ultimate challenge in transplant medicine. Unlike hearts or kidneys that operate in relatively closed systems, lungs are constantly exposed to air, bacteria, and the outside world. They have massive surface areas packed with immune cells that attack anything foreign. That's why lung transplants have historically had the worst success rates of any organ. Today, we'll explore how Chinese researchers overcame these seemingly impossible biological barriers, what the 9-day timeline actually tells us about the future of xenotransplantation, and why you might someday owe your life to a genetically modified pig. The pig used in this experiment wasn't ordinary. This was a Bama Xiang pig that had undergone 6 precise genetic modifications using CRISPR technology. Think of these modifications as molecular diplomacy, teaching pig cells how to speak the language of human biology. 3 pig genes were completely knocked out: GGTA1, CMAH, and B4GALNT2. These genes produce molecules that human immune systems immediately recognize as foreign invaders. They're essentially giant, not human signs painted on every pig cell. Remove these genes, and you eliminate the most obvious targets for immune attack. But removing the rejection signals wasn't enough. The researchers also added 3 human genes: CD55, CD46, and THBD. These act as molecular ambassadors, telling the human immune system that this pig organ is actually compatible. CD55 and CD46 help control complement activation, which is part of the body's rapid immune response. THBD helps manage blood clotting, preventing the dangerous clots that often form when pig and human blood interact. This combination, known as the 6GE modification, represents years of trial and error to identify exactly which genetic changes would create maximum compatibility. It's not random editing. Every modification addresses a specific biological incompatibility between pigs and humans. The precision required for this work is staggering. We're talking about editing genes in pig embryos, raising these animals in sterile environments, and ensuring every cell carries exactly the right modifications. The pig that provided this lung was essentially a living pharmaceutical factory, engineered from birth to save human lives. Now let's walk through what actually happened during those 9 days, because the timeline reveals both the promise and the remaining challenges of this technology. Day 0. The surgical team at Guangzhou Medical University transplanted the genetically modified pig lung into the left side of the patient's chest while keeping the right human lung intact. This allowed them to directly compare how the pig lung performed alongside native human tissue. The first 24 hours were critical. In previous attempts at pig-to-human transplants without genetic modification, organs typically failed within minutes due to hyperacute rejection, a violent immune response that destroys foreign tissue almost instantly. But this time, something different happened. No hyperacute rejection occurred. The pig lung was accepted by the human immune system. However, at 24 hours, severe swelling developed. This wasn't immune rejection. This was ischemia-reperfusion injury, the same complication that affects human lung transplants. When an organ is removed from one body and placed in another, the period without blood flow, followed by restoration of circulation, causes inflammation and fluid buildup. Days 3 and 6 brought new challenges, episodes of antibody-mediated rejection. The human immune system had time to analyze this pig lung and began producing antibodies specifically targeted against pig proteins. These antibodies triggered inflammation and threatened organ function. But here's what makes this experiment remarkable. Each time, the lung recovered. By day 9, when the family requested the experiment be ended, the organ was showing signs of partial recovery from the latest rejection episode. 9 days might seem brief, but in the context of xenotransplantation, it represents a quantum leap. Previous pig-to-human organ attempts failed within hours or minutes. This lung sustained human life for over a week, proving that the fundamental biological barriers can be overcome. The patient received an immunosuppression protocol unlike anything used in conventional transplant medicine. We're talking about 8 different drugs working in combination: ATG, basiliximab, rituximab, eculizumab, tofacitinib, tacrolimus, mycophenolate, and steroids. This cocktail was designed to suppress multiple pathways of immune response simultaneously. Some drugs targeted T cells, others targeted B cells and antibody production. Eculizumab specifically blocks complement activation, while tofacitinib inhibits inflammatory signaling pathways. The intensity of this regimen highlights both the potential and the problems with current xenotransplantation. Yes, you can make pig organs work in humans, but the immunosuppression required is so extreme, it would be toxic to living patients. The drugs that kept this lung functioning for 9 days would likely kill a living person through infections or organ toxicity within weeks. This is why researchers emphasize that we're still years away from clinical trials in living patients. The genetic modifications need to be refined to reduce immune response, and entirely new approaches to immunosuppression need to be developed. Some promising research focuses on teaching the immune system tolerance rather than simply suppressing it. This lung transplant is part of a broader xenotransplantation revolution happening right now. Pig-to-human kidney transplants are already in early clinical trials with living patients at hospitals like Massachusetts General. Heart transplants have been attempted in compassionate use cases for patients with no other options. But lungs were always considered the final frontier because of their complexity and constant exposure to environmental threats. The fact that this barrier has now been crossed suggests that xenotransplantation is moving from experimental curiosity to potential clinical reality. Companies are taking notice. eGenesis recently raised $191 million specifically to commercialize genetically modified pig organs. Other companies are developing competing approaches, including different genetic modification strategies and alternative immunosuppression protocols. The competition is intensifying as everyone realizes we're approaching a tipping point where animal organs could become routine medical treatments. The question is no longer whether this will work, but who will bring it to market first, and how quickly safety concerns can be resolved. However, significant challenges remain beyond just making the organs work. Long-term safety monitoring is crucial because pig organs could theoretically harbor viruses that jump to humans. The CDC requires that xenotransplant recipients be monitored for up to 50 years, with biological samples preserved for future testing. There's also the question of scalability. Even if the technology works perfectly, producing enough genetically modified pigs to meet global organ demand would require massive agricultural and manufacturing infrastructure that doesn't currently exist. This breakthrough raises profound ethical questions that society will need to address as the technology advances. Using brain-dead patients for experimental procedures requires careful ethical oversight and genuine informed consent from families dealing with tragedy. There are also questions about resource allocation and equity. Xenotransplantation will likely be expensive initially, potentially creating situations where wealthy patients can access unlimited organs while others wait for scarce human donors. From a global perspective, different countries are taking varying approaches to xenotransplantation regulation. China has moved aggressively forward with these experiments. The United States has established extensive safety protocols, but is proceeding more cautiously. European countries are still developing regulatory frameworks for clinical applications. The cultural and religious implications also vary significantly across societies. Some communities may have objections to receiving animal organs, while others may embrace the technology as life-saving. But perhaps the most profound ethical question is this: if we can eliminate the organ shortage through genetic engineering, do we have a moral obligation to do so? Every day we delay bringing this technology to clinical reality, people die from preventable organ failure. So when will pig organs start saving actual patients? The timeline varies significantly by organ type and depends on overcoming different technical hurdles. For kidneys, we're closest to routine clinical use. Living patients have already received pig kidneys in early trials, and the results are promising enough that larger studies are being planned. We might see kidney xenotransplants becoming available for select patients within 3 to 5 years. Hearts are more complex, but compassionate use cases have already been attempted. With refinements in genetic modification and immunosuppression, heart xenotransplants could be available for end-stage patients within 5 to 7 years. Lungs, as this experiment demonstrates, are the most challenging. The inflammation and rejection episodes seen in this 9-day study show that significant optimization is still needed. Experts estimate that lung xenotransplants for living patients are probably 7 to 10 years away. The path forward requires several key developments. First, the genetic modifications need refinement to reduce immune response and improve compatibility. Second, immunosuppression protocols need to become less toxic while remaining effective. Third, organ preservation methods need improvement to prevent the inflammatory damage seen in the first 24 hours. Manufacturing and regulatory approval will also take years. Producing medical-grade genetically modified pigs requires specialized facilities, extensive quality control, and regulatory oversight that doesn't currently exist at commercial scale. This 9-day pig lung experiment represents more than just a medical achievement. It's a glimpse into a future where organ failure no longer means death or years of waiting for a compatible donor. Imagine walking into a hospital with end-stage organ disease and being told that a genetically compatible organ will be available within days, not years. That's the future xenotransplantation promises. But this future isn't guaranteed. The technical challenges are formidable, the regulatory hurdles are complex, and the economic barriers are substantial. Success will require sustained investment in research, careful attention to safety, and thoughtful approaches to ethical concerns. The genetic engineering techniques that made this pig lung work are improving rapidly. CRISPR technology is becoming more precise and powerful, allowing for more sophisticated modifications that could further improve compatibility. Alternative approaches are also being developed. Some researchers are working on regenerating human organs from patients' own cells, which would eliminate rejection entirely. Others are developing mechanical devices that could replace organ function. But xenotransplantation has unique advantages. Pigs can be bred in unlimited numbers, genetically modified to exact specifications, and raised in controlled environments to minimize infection risk. They're also anatomically similar enough to humans that surgical techniques translate well. 9 days of a pig lung functioning inside a human chest. It sounds impossible, but it just happened in a hospital in China. More importantly, It proved that the biological barriers that have separated animal and human organs for millions of years of evolution can be overcome through genetic engineering. This isn't the end of the organ shortage crisis, but it's the beginning of the end. The technical proof of concept now exists. The genetic modifications work. The surgical techniques are established. The regulatory pathways are being developed. The remaining challenges are significant, but not insurmountable. Better genetic modifications, safer immunosuppression, improved preservation techniques, and scaled manufacturing are all solvable problems given sufficient time and resources. For the millions of people currently waiting for organ transplants, this experiment offers something that's been in short supply: hope. Hope that their wait might be measured in weeks rather than years. Hope that organ failure might become a temporary setback rather than a death sentence. The patient in Guangzhou was already gone, claimed by a brain hemorrhage that couldn't be treated. But for 9 days, a part of a pig kept human lungs breathing, human blood oxygenated, and human life sustained. In doing so, it showed us a future where the phrase, "compatible donor not available," becomes extinct. We're witnessing the early stages of a medical revolution that could be as significant as the discovery of antibiotics or the development of vaccines. The organ shortage crisis has claimed millions of lives over decades. This 9-day experiment suggests those deaths might not have been in vain if they led us to this breakthrough. Thanks for joining us on episode 18 of Curiosity AI Podcast. If this exploration of the intersection between genetic engineering and life-saving medicine intrigued you, make sure to subscribe for more analysis of the scientific breakthroughs that are quietly reshaping human survival. Until next time, remember that the future of medicine isn't just being discovered in laboratories, it's being genetically engineered to save lives.